insightful comments. This is a huge topic that we face, and we'll be facing it well into the future. And I think the more we discuss this openly and in our academic environments and take back the ideas that are discussed here, the quicker we will come to evolve into a system which recognizes these important events in the academic community. So to continue with Session C, and particularly in highlighting the significant impact NAI fellows and members have contributed to the betterment of society, our next speaker will be Mark Humayun from the University of Southern California, who's going to be talking about advanced implants for ophthalmology.
Okay, great. Let's see if that works. Okay, I think maybe we can switch to my computer. I'd like to thank the NAI for the opportunity to present today. Let's see, why isn't that? Let's see, for some reason. So today, let's see if I can get this to work. We tested it earlier, but anyway, I'll be talking about advanced implants for ophthalmology. And I'm a professor of ophthalmology at University of Southern California, and I have a PhD in biomedical engineering. And I split my time, half-time in the clinic and half-time doing research to develop these technologies. So, sorry, it's not. Anyway, I'll be talking about 3 different technologies for the eye, and really they have a lot of the discussions that we just had were very relevant because being in the medical setting and being in the university, it was very challenging to get these technologies to be commercialized. And one of the things that really helped me through the process were not only the students, which were incredibly helpful and always pushed the ideas forward, but also the ability to have startup companies form really early on. And Bob Greenberg is in the audience. He's the CEO and president of Second Sight, one of the companies that developed these technologies. And so he really, having those sorts of companies be with us really helped us to develop this technology and commercialize it. So with that, you know, let's see. Okay, so basically, if we could go to the next slide then. Great, thanks. So, you know, the eye is a very difficult place to implant devices. It's very small, it's mobile, it's very delicate. But the first intraocular lens implant, believe it or not, was done in 1950 by Sir Harold Ridley, who had actually noticed a very interesting phenomena. The canopy from World War I fighters, airplane fighters, when they were shot down, sometimes went into the eye of a pilot, and he had the foresight of looking into that and realizing that you didn't need to remove, actually caused more problem removing the canopy, the piece of plastic from the eye, and he actually left it in there, and the eye did well. And so this serendipitous finding actually led him to put and develop an intraocular lens implant in 1950s. Just to give you an idea, the first pacemaker was 1956. So the eye actually, one wouldn't think so, but was one of the earliest places to implant devices. So today I'll be talking about things that have advanced way past that. Retinal prosthetic, a chip that actually restores sight to the blind, again, as I mentioned, in collaboration with Second Sight, some drug delivery implants, and possible future electronic devices. And this picture down here shows you a little contact lens that Google is developing. And it's very interesting to see not the traditional medical device companies be involved, but others outside that getting into this space. And this is for measuring, for example, glucose and perhaps doing some other things as well. And then I'll conclude with scaffolds for cell-based therapies. So the first technologies I mentioned is to restore sight. It was really an idea to IPO. The company is now formed, and basically the device functions as such. It has a tiny camera in the glasses which captures the images. And once these images are taken and converted into a wireless communication methodology, so it's an inductive coupling. It takes the data and power, so there isn't a battery inside the device. Actually, power is also being transmitted from outside. So both data and power are then sent to a device that's implanted in and around the eye, and then this implanted part of it decodes this information, both, again, power and data, and then sends it to the inside of the eye, the retina. So the retina, the reason most of these patients are blind because their photodetectors or photoreceptors are damaged. So what this device does is effectively stimulate the remaining nerve cells and jumpstarts the otherwise blind eye and sends the information to the brain. So for example, this letter E would be then conducted through tiny electrical pulses to stimulate the remaining nerve cells and sends the information to the brain. So that's how this device works. As you can imagine, it was clearly science fiction when we got started, and now it's science fact. This is the device, the Argus 2, which is now approved in Europe and in the US. As I mentioned, manufactured and distributed by Second Sight Medical Products. It has this video processing unit, which you didn't see in the animation, which is either worn on a belt or on a strap, converts the information, and then sends it through the telemetry unit that I did show in the animation. So here's a patient, if we could turn up the audio, who's been blind for 30 years, who's using this device.
Yes.
And this device only has 60 electrodes. And one of the main questions was, in the eye there are hundreds of millions of photoreceptors. What is the chance that
smaller?
Yes.
what is the chance that 60 electrodes could actually restore any sight?
Yes.
So you can see this patient again, completely blind without this device,
Yes.
is looking at it, and at the bottom you have the camera.
Okay.
Yes.
See?
Yes.
I know.
Yes.
So this really changed the perception around the world that your eye, a very high resolution detector, can actually be interfaced with something that has 60 channels of information, and that's taking this information from the glasses and a camera in the glasses that's worn. So what's interesting is that this is not where we've gotten. Obviously, the letter size is the size what fits in your palm, arm length away. With zoom, with 16x zoom, we've seen that in some subjects and some patients we can get up to 2200. So what is 2200? That's the big E at the end of the room. So we still have a ways to go, but at least this technology hasn't hit its limit. Let me talk a little bit about color vision. It's very exciting because this helps provide content information at lower resolutions. You would think, well, why do we need color? We really don't need it, but let me make a case why we do need it. For example, if you look at these two objects at low pixels, low resolution, you wouldn't be able to tell what they are. You can kind of tell they're in the shape of a ball, but if I gave you this information, you might say, now I see. Perhaps one is an apple and the other the orange. So you can see how color does have information. And similarly in this path, you can see if they could tell the green grass and the fall trees from the pavement, that it provides useful information for mobility and orientation. But the problem is that we cannot possibly get onto the red and green and blue pathways. As I just mentioned, we only have 60 electrodes, and they're very big and almost to the size of 20 of each of those photoreceptors, if not bigger. So how do we do it? And one of the interesting things we're doing is when you can't do it in spatial domain, you can try to do a frequency modulation. And this is a little test. When I was a kid, I used to spin what's called a top, which is a black and white top. But at certain frequencies, you would see these various colors at certain frequency of spinning the top. So that gave us the idea, well, could we actually use frequency encoding to try to get some colors? And as you can see here, this is a little bit of data showing that we can get up to 8 colors by using frequency encoding. So it's a very interesting area to develop further. Obviously, a lot more work to do. We've done a lot to get it here, but a lot more work to do. And again, it was really through getting students to be part of this. We've had more than 200 students be part of it, multiple universities where I was at, and also who collaborated, and our corporate partner, which ultimately enabled this to happen. So it was a combination of federal as well as private funding. I'll speak a little bit about a drug delivery pump. And here the issue is that, as I mentioned, as an ophthalmologist, I treat many patients with retinal problems, and they're wonderful drugs that are protein or antibody-based, but it ends up requiring every month injections into the eye. And so you could imagine, I tell Mrs. Jones, the good thing is your macular degeneration, we're able to treat it. The bad thing is that you need an injection every month for the rest of your life. So there are a number of different types of drug delivery devices, but they're challenged when you try to develop protein or antibody drugs into the eye. There's lots of wonderful work being done all around the world on how to do this. And what we've developed is, again, a small device that goes around the eye that in a 15, 20-minute procedure can be implanted and thereafter refilled in the clinic to deliver drug into the eye. The big challenge was, I think some of you are familiar with these large Medtronic pumps. They fit into your body and they're very big. And how do you miniaturize this such that the pump can actually fit around the eye, and then you can take drug directly from a vial and inject it into the pump and have it deliver into the eye. Here's a little bit of data and to show that we targeted 8 microliter delivery, and it's very accurate between plus or minus 20% over 60 approximate doses. So here's a little video. Now you're looking into the eye with the little endoscope camera, and you can see we loaded the pump up with a fluorescein dye, and it's coming into the eye. It's very important not to inject a lot of drug quickly into the eye because obviously it can elevate your eye pressure and cause damage. So it's a very controlled delivery into the eye. And another example, we advanced this to phase 1 studies in 11 subjects with diabetes. We published this at the bottom for those interested in the reference. And so far it's going well, no serious or unexpected adverse events. One of the things we were worried about is, does this cause double vision because the pump is sitting on the eye and so forth, and it really has not. It's been a wonderful technology that continues to be developed. And the last bit is about how do you take these technologies and actually blend them with stem cells. And so here you can see that there are 2 types of macular degeneration. The wet, I already mentioned, the bleeding type where it does need injections, but the dry type, which is you might have heard about, really requires a monolayer of stem cells to be implanted. And I'll just speed forward to show you that in order to do this, we had to develop a synthetic basement membrane. Here's the device relative to a penny that grows these RPE cells on them. And I'll skip the surgery and just show you that we're able to put this. This is an image into the eye. This is the retina, and you can see the well-formed monolayer of the cells that we've been able to implant in patients in a phase 1 IND, whereas if you just inject the cells without this basement membrane, synthetic basement membrane, they don't form in a proper manner. So basically, to conclude, there are lots of issues related to biocompatibility in the eye that are very unique, and I've tried to highlight them a little bit to you. Bioelectronics and nanoscale scaffolds require a very safe and long-term abiotic-biotic interface for them to work. And of course, various technologies as we develop them will be enormously helpful. So I'd like to acknowledge this Argus 2 study group, which is helped with the retinal prosthesis, the drug pump group, which helped with the drug development, of course, the stem cell patch group, who helped with the stem cell technology. And lastly, as many of you mentioned before, and the panelists did too, a lot of the funding does come from federal as well as state, but also corporate, as I mentioned, is very critical. And I'd like to leave you with the patients here at the FDA panel review, who were really critical to get the technology through. So I think we talk a lot about intellectual property and what we do and how to file it, but the final step is to actually commercialize it and get it approved. And you can't really do it without the help of these subjects who are really very critical. So thank you for your attention.
Talks have just been like out of dreams. Amazing technologies. So I'm very pleased to bring up to the podium Hani Farid, who from Dartmouth College is going to talk about digital forensics.